Fuck. Yeah, good catch. Right, let me go back. No, 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 no. Back, back. Right, okay. I've just started recording. Thank you so much, Jan, because I completely forgot to completely forgot to press record. Just so you know, none of you guys will be on the recording, so I can't see your faces or anything like that. But I have this is the you'll be able to watch the video back at least. Um, so the next sort of section we're going to talk before I start doing the agar stuff, I'll probably talk to you about sort of clean working. Um, we someone asked earlier what a flow unit is, so I'm going to keep briefly tell you what a flow unit is. But most of you will be more accustomed to working in a still air box or just out in open air with inoculating in just out on a kitchen surface or whatever. We're going to talk a little bit about vectors for contamination, and I've put a little list here of you're breaking up. Um, I'll try and take things a bit slower, and that way, if there is any audio, if I freeze at any point, just ping me in chat, and I can redo it. Saying that, I think this is recording locally for me as well. So if you do miss a little bit, it'll be recorded my side anyway. So you'll be able to watch it back after. So is anyone else using your internet? Yeah, my daughter's upstairs playing games. So um, I think that the recording the other side of this should be fine. So if you want to rewatch it after and there's any lag. No, I've got a 200 meg internet connection, but yeah, I mean, you never know. Let me just check that I am on the right. I'm going to just stop sharing 1 second. I'm going to just check I'm on the right internet. Give me 1 minute. Stop sharing. No, I am on the right internet. Share screens. Share. Right. Chat. Yeah, I know I'm bragging about my internet speed and then having it still freeze is a bit of a right. So once we've talked through agar, I'm going to kind of take you a little bit outside of that, and we're going to go into liquid cultures. We're going to kind of circle back to liquid cultures and how that relates to agar, and then finally I'm going to use one of our beginner grow kits to demonstrate grain inoculation and talk to you a bit about that process onto substrate, and then finally onto fruiting. Um, I'm going to try and keep it really beginner friendly. I know there's varying experience levels in the chat, so I understand to some of you this is like, I'm just flogging a dead horse or getting you to suck eggs or whatever the right phrase is. And right at the end, we're gonna do a Q&A. So right, I'm gonna just jump on a little bit now then and go on. I've got a few more slides that just kind of demonstrate well a little bit about sort of mushrooms in the wild. For those of you who don't go outside very much or and you don't see mushrooms in the wild. This is the reproductive cycle of a mushroom. So mushroom in the center, when the conditions are just right. So often in autumn, the air pressure is changing. Um, the weather becomes cooler. There's more airflow. Mushrooms tend to use that as an opportunity to disperse their spores. Spores come from these tiny little microscopic bits called basidia. So all the cap and stem mushrooms, well, almost all of them are in a family called Basidiomycota, and that includes a bunch of other bits as well, like all the boletes and penny buns, all that sort of stuff. Um, they all have these little things called basidia, and inside each one of these is, I think, 8 spores for Basidiomycota. This sounds technical now. Um, so as the spores are released, they'll land on a suitable substrate. So it could be a piece of like it could be in the soil, it could be on a piece of wet wood. The spores will germinate and they send out little threads called hyphae, which most of you guys are going to be familiar with. They're these tiny little sort of unicellular threads that they head out. They're looking for both a compatible hyphae to mate with, and they're also looking for different food sources. So they're always exploring in different areas for different food sources. When two compatible hyphae meet, they create a thicker strand of called a mycelium or mycelium as it's referred. And that what we're going to do as cultivators, we're trying to kind of first get to this stage where we have mated we have mated hyphae, and this process here, where the two hyphae have mated, that's where mushrooms are born. If you just had hyphae, say you just had a single spore land on some agar and it grew out, it may look like mycelium, but it'll never produce a fruiting body, a mushroom fruiting body, unless it's met a compatible mycelium. Once the mycelium once the mycelium is covered the substrate and the conditions are right, it'll start to form these little hyphal knots. So you'll see this for those of you who've ever grown, for those of you who have grown, well, anyone who's grown anything, I guess, on the surface of substrate, you will sometimes see these little white balls, and they effectively will be what becomes the mushroom. They're little storage. I think from what I can tell, they're responsible for storing nutrients and water just in case the conditions change. So it allows the mushroom to still continue to be developed even if like the weather changes suddenly. So they're almost like little storage facilities um, for nutrients and water. That will then change into a primordia, which is a tiny little mushroom, like what we refer to as pinsets in cultivation, which is just like a tiny little baby mushroom, and eventually that will become a mushroom again. And then the cycle starts again. So, once the mushroom reaches maturity, it releases spores, blah, 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 blah. This is going to be something that some of you are quite familiar with because you may have grown mushrooms before. You may have bought mushrooms from the supermarket and seen spore deposits in the bottom of the packet. So that's exactly the same thing. It's the conditions were once it's been picked, it's the mushrooms reach maturity and it's desperately trying to produce spores to continue its genetics. Um, so where do spores come from? You'd be shocked to realize that spores do come from a mushroom. They don't just come from friendly spore vendors. So here we're going to focus on obviously one that some of you might be familiar with. This is a Psilocybe cubensis. And you can actually see here, just on the this part here, there is on the veil, which is the little bit that's dropped away. You can actually see spores deposited on there. There's like a purple brown ring that goes around the bottom of the mushroom. I put a little photo in of spores under the microscope. Again, this is some Psilocybe cubensis spores. And so the way we normally would collect spores if you found a mushroom in the wild, you can effectively just pick the cap, put it under you can put it on foil face down, so the spore side down. You put a cup over the top, and I normally like to just put a little drop of water on top of the cap, and then that will sort of create the right humidity, and also the fact that the mushroom knows that it's been disconnected from the wider organism, so this is its last opportunity to spread its genetics. So it will drop its spores shortly after, and there you go. You can see a beautiful spore print, developing below the mushroom cap. And then the spore print there, that's a really the bottom right is Psilocybe cubensis spores on foil, which is often they're sold as spore prints on foil for cultivators to work with, obviously within countries where it's legal. Yeah, so that's kind of that's a quick overview of spores, and I guess what we do as spores come from orangutans. They can do sometimes, yeah. I'm going to stop sharing and I'm going to switch over to my other camera. Do, 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 do. Oops, that's not the way to do it. Share, advanced, second camera. Okay, you guys should be able to see my second camera. Can you see the FaceTime stuff here? Yeah, and you can also see me as well, like in a little window. Okay, cool. Maybe have a quick sip. There was a question just came up actually. I'm just going to see. I have grain spawn, but it's brown stuff at the bottom. Joe, I'll definitely answer that at the end. We're going to cover like contamination stuff, so I'll lump that into that section. Right. Let me just make sure I've got my list of bits that I'm going to cover in front of me because I'm not that well versed in this that I don't need a list of stuff to read. Right. So as cultivators, we're trying to recreate that sections of that life cycle of the of mushrooms growing from a substrate, but we're removing a lot of the factors that you have in nature. So, whereas when a mushroom or mycelium in general starts to develop in nature, it has to compete with different spores from different mushrooms. It has to compete with molds, yeasts, insects, human beings, chemicals. All these different things occur in nature that could prevent that mycelium from ever creating mushrooms. So what we do as cultivators is through sterilization and clean technique, we isolate just the sort of the mushroom genetics, and we try and work with that in isolation from all the other things that might damage the process of producing fruiting bodies. So what we're going to do is I'm going to take you through first kind of the process of getting to liquid culture. So some of you might be familiar with spore syringes, which is so spore syringes are effectively they're sterile water with we saw the spore print earlier with foil. People effectively scrape the spores off the foil into sterile water, which looks, I guess, a bit like that. Um, they suspend the spores in that liquid solution, and they draw it up with a syringe. And you can buy these syringes from different spore vendors, or um, so they're typically, the spore syringes typically are used by people selling cubensis spores or magic mushroom spores, as that's the only sort of legal format that you can currently buy them in. Because they're not germinated and they don't contain any psilocybin, they're still technically legal. They're not legal to cultivate. It's not something that we suggest you do with our products. Um, I think there are some people in different countries where it is legal, like Austria, where we send a lot of stuff. Um, they obviously can use our products to grow psilocybin cubensis, and they will grow just the same as any other mushroom in the same process, pretty much as what we do with gourmet growing. But the process I'm going to take you through tonight obviously is not is not going to be anything at all to do with cubensis, so don't get your hopes up. Um, right, so I'm going to show you how to introduce mycelium to agar first. So I'm going to bring down a stack of agar plates. I'm going to put my gloves on, and I'm going to show you kind of how we get from a spore syringe, for instance, first, which I'm just going to use as like a placeholder, like a syringe that doesn't contain spores. It contains liquid culture, but I'm going to use that as an example. Like I said, I've got this flow unit in front of me, which you can see here. It's not on, but what I wanted to do was show you, you know, how in a professional setting you would work in front of you'd work in front of a flow unit like this, and it basically produces clean air out the front of it. It draws dirty air in the back, and there's a paper filter on the front here. It forces HEPA filtered air out the front, so it gives you this zone to work in where you effectively have a clean stream of air running in from this direction outwards. So there's a principle in mycology called first air, which is, if I have something dirty here and I'm working over here with something clean, as you can imagine, the air is going to flow over it and it's going to end up in the clean thing that I'm trying to work with. So normally we would have when we're working with different things, if I'm, for instance, doing a clone with a mushroom, which I'm going to show you in a minute, we wouldn't sit a dirty mushroom straight out the bag here and then be working here like this because it would contaminate. There's a funny lag between cameras, just so you know, to fix later. Yeah, I'm hoping that this will all record properly. Let me just see how my hands move like that. They seem right this side, but I'll try and take things a little bit slowly. I knew there would be some technical issues tonight, but hopefully the lag isn't like, as long as it's not like 20 seconds or something. Top down, very laggy, like 3 frames per second. Okay. Um, that's a bit annoying. What I might do is I might just I'm going to turn off my main camera. I'm going to switch it to the, I'm going to switch it just to the top down camera. And then I'm going to turn off sharing because you don't need to see my face anyway. So, uh, content only. Oops. Okay, can you just see one camera now? Yes. Okay, cool. Right. I'm hoping that I think what it might be is that obviously it's technically trying to get two cameras to run in run in tandem is a bit of a pain. Hopefully things match up now a little bit better and you can you know, we won't just have this big gap. Um so right, we're gonna talk about agar. So what is agar? Um so agar is basically it's these are sterile petri dishes. Um and they have a nutrient-rich gel on top. We've created these in our lab. Oop, Jesus Christ. Should have just taken the wrapper off beforehand. So we have a stack of plates, they're all vacuum sealed. So inside here is agar, which is a gelling agent, and it also has malt extract in there, which is basically like a nutrient food source for mycelium. It's really simple carbohydrates, but it also has some other like good nutritional things in there that mycelium tends to really like, and will sort of all correspond to having a good, healthy organism. I'm going to take these out. So, fresh, brand new scalpel, stolen from MycoPunks Limited. Right. Right. So, um, before I start, I'm going to just talk a little bit about obviously clean technique. I said I'm working in front of a flow unit here. Um, what most of you might be akin to is a still air box. So, a still air box is basically a plastic tub with armholes cut in it, and it what it does is you disinfect the inside of the box. And it's basically like like a really useful tub type box where you can see into it, the sort of thing you'd store old bits in. Um, you cut some holes in the front that will allow you to work inside the box after it's been fully disinfected. And the point of a still air box is that once you've disinfected the inside, you're trying to disturb the air as little as possible. So you're working slowly and methodically with all disinfected items. Um, and the point is that the air in the room is still has bits floating around in it, but the inside of your still air box has still air. So anything it won't introduce new contamination, and it effectively gives you this little working space to work in that allows you to do clean work. Obviously, certain things are more difficult than others for if you're doing if you're working with larger things and that have potential to disturb the air or move too quickly, that can cause problems in a still air box. Um, in a professional setting, we use flow units because it allows us to just work, in front of a big wall of clean air, and it makes your workflow so much more simple. Um, I'm gonna after we finish, I will put in the chat a guide on how to make a still air box. So if you guys are absolutely new and you want to do some of these techniques, at a minimum, you're going to need a still air box. Agar specifically is the second I open this up now in the room, there's airborne contamination everywhere. There's yeasts, there's all sorts of mold spores and stuff that are always flying around in the air. Um, normally if you're starting any sort of clean work, you'd make sure that there's no airflow in the room, even if you were working with a professional flow unit. You would close all the doors and windows and ensure that there's no external air blowing, creating like a bellowing effect of air. Um, so everything's vacuum packed in here. The second I open this now, obviously we're in air, but pretend I'm working in front of a functioning flow host that's on and plugged in. So one of the cool things about these packs that I should probably tell people that I never actually do. Um, so obviously they're vacuum packed at the moment, so they're still sterile. People often say to me like, "Oh, can't you do these in packs of like five because I only use five at a time?" What you can do is you can run your scalpel just along the top here like this, spin them round, keep spinning, keep spinning, keep spinning, and then you can just take off one at a time. So you can actually, that now is still all sealed. And so you can literally say, right, I know I'm going to use three. So you could just cut away three and then have that just as like remains relatively sealed. Um, so we've got our agar plate. I'm going to open it up just so you guys can see it. Um, This one, this is from our little Halloween pack that we did. So we've done like bright green. We've added food coloring to these, so just to make them a bit more interesting. The food coloring doesn't offer any benefit other than, it can look cool when the mycelium's growing on it. So in this one, we've got two different mixes. We've got a black one and a bright green one. And it really does come down to preference. There's some evidence to show that certain food colorings, could affect the mycelium growth. And that's something to be aware of when you're cultivating. If you're worried about uh, E numbers and things like that, generally speaking, mushroom mycelium is going to break all of that down anyway and metabolize it, so it isn't going to be an end product. So that's the agar gel there. And what this does is it effectively gives us a two-dimensional plane, which is the surface of the agar. When we introduce spores of mycelium, that mycelium is going to grow just in one direction, and it's going to grow out across the plate. And I've got some plates over here that I'm going to show you. So, got a very over-colonized. This is an oyster mushroom. So, there you go. You can see there's a bit of contamination on there, so you might be able to see where the mycelium's not touched. Um, what it is there was a bit of this is actually from a cloned mushroom that we introduced. So we introduced a small piece of tissue from a wild mushroom, and I'm going to show you the process of that in a minute. Um, so when we introduced it, there was a small amount of bacteria on there, and that ended up creeping across the plate here. And what you'll see when you're growing mycelium on agar is that um, it creates sort of little zones where the mycelium will grow, and if there's anything else on the plate, it will just refuse to grow there, or if it's a weaker organism, it might just steamroll straight over it. Typically, if we see issues with contamination on agar, we would try and isolate the good mycelium and move it to the next plate. So yeah, that's what mycelium looks like. This is an overcolonized plate, and there's two types of the mycelium that we typically see in terms of growth patterns. Let me just see if I can grab another one. Ooh, got. I'm going to show you some rhizomorphic growth. So fluffy growth, like you'd expect from an oyster mushroom. And then here we've got an example of rhizomorphic growth. So if you look at the center there, um, it's basically like this ropey growth. This has had some issues with bacterial contamination, but as well, which is why the growth is kind of changed towards the outside of the plate. But you can see those beautiful sort of ropes, and this is something um, uh, that a lot of psilocybin growers specifically, they use this as an example of like an aesthetically pleasing plate. Um, Psilocybin mushrooms do tend to create, in most cases, will create rhizomorphic mycelium if the conditions are right. So you get this really beautiful pattern. This is a reishi species from Barbados. So there you go. You can see that growing nicely across the plate. So you got the two different types of mycelium there, rhizomorphic, tomentose, which is fluffy, and then rhizomorphic, which is these beautiful ropes. Um, People used to say that rhizomorphic growths would produce better results, but it looks like there's no evidence to show that. So sometimes you might be growing a mushroom where you expect good rhizomorphic growth, but it doesn't tend to have any effect on the end product. It can be down to a number of things, and sometimes the mycelium will just change suddenly from being rhizomorphic to tomentose. It could go from tomentose to rhizomorphic. But one of the good things about rhizomorphic growth is when we looked at the diagram earlier, I talked about hyphae being sent out. Um, when it becomes mycelium and it meets a compatible mate, it creates thicker mycelium. So with the hyphae here, with the mycelium growing out here, with this rhizomorphic growth, that's an example of mated mycelium. You just don't get growth like that where you have these thick strands of mycelium when it hasn't mated. So this mycelium here, if we were to transfer this to grain, it would definitely produce fruiting bodies because it's mated mycelium. It's ready to it's basically it's found the love of its life, and it's ready to have kids. Video quality going down. What a nightmare. Um, for those of you who like, where the video quality is flaking out, and I know few new people have joined, um, this is all being recorded locally on my device as well. So I'm hoping that the video quality after when I publish this will be a lot better. Um, so how do we go from um, how do we go from this blank agar plate to growing out mycelium? Yeah, exactly. Good luck with that, Jan. I don't think, getting my daughter to stop playing Roblox, it ain't gonna happen. So we'll take the wins where we can get them. Right. Okay. So how do we get mycelium on the plate? Let's start off with spores. Right. This is not actually a spore syringe. This is a sporeless oyster. So there's actually liquid mycelium in here, but most of you are used to seeing well, most of you. I'm not making any judgments here. Some of you are used to seeing spores suspended in a syringe, and like I said earlier, they come from the underside of the mushroom. They're scraped into distilled water, and then they're suspended in a syringe like this. Often they're really difficult to see as well. So like if you do buy a spore syringe and you're like, there's hardly anything in here. If you hold it up to light, you normally can see a lot better. So if you hold it up to a light source, bear in mind they're pretty much microscopic. So and also you might notice sometimes with spore syringes that the spores are all clumped up in like a little ball. That's because they carry a small electrical charge and they're all drawn to each other in the liquid. And what we normally do, even whenever we're working with syringes, we tend to like really want to disturb things up before we use them. So I just shake things side to side like this. Um, and what that'll do is that will just disturb the inside liquid. It'll break up that spore clump and allow you to, have a better distribution of spores throughout your syringe. What we sometimes end up doing is you might inoculate something with some spores, and from where they've all clumped up into one area, you might end up with one really good grow, and then just nothing produced in other bags. So that can sometimes be from where your spore syringe just hasn't been shaken up enough. Um, so what is the difference between a spore syringe and liquid culture? So a spore syringe is not a sterile product yet. The process I'm going to take you through now will the outcome of it will be a sterile product that you can use and be confident that you're not going to have contamination in your grows. Other as long as other sort of thing, processes are followed correctly. Um, so if you are working with spore syringes and you're just inoculating straight into a bag, you've got to think about the process that has taken that to get your spores out of the mushroom that they've been put face down on your foil. There's other airflow and stuff that's come in. This mushroom's already got contamination on it. So the source of the spores is never going to be perfect. There are going to be other spores that have landed, in the gills, unless someone is growing, the original mushroom in a sterile environment, which just doesn't really happen. You will have other competing spores in with the spores that are gathered from a mushroom in a spore syringe. So, if you've ever sort of bought from a spore vendor before and kind of had trichoderma or any issues with contamination around yeast and things like that, it's a difficult conversation to have because it's expected that there will be those sorts of contaminations in with there's only so much we can do um, to to completely remove contamination vectors when you're creating a spore syringe. Um, Ultimately, it's non-sterile spores in a sterile solution. So the water itself is sterilized and clean. It's done in front of a flow unit. The spores are scraped off into the liquid in a very clean environment. Everything's done as best it can be, but the spore, the source of the spores is still not a sterile product. So what we're going to do is I'm going to show you how we would inoculate agar with spores. Like I said, use your imagination. This is liquid mycelium in here. So you don't need a needle for a start, really, um, for inoculating agar. And you're looking for really the tiniest amount of liquid on the plate. If you flood the plate completely, you'll struggle with you'll just basically create a too wet an environment for the spores to germinate in a nice way. What you can do if that happens is you can effectively just leave it a lot longer, and as the plate dries out over time, you'll probably find it will germinate. But we're really looking for the smallest amount of of liquid that you can get on this plate as possible. This is quite a finicky thing, right? Because these syringes, when they sit for a while, the plungers get a little bit stuck. So what I sometimes like to do is with the cap on, before I go to do this, I give like the syringe like a little pump, just to kind of get the plunger moving. And that way you won't get this immediate like as you're trying to put the spores in, you won't just suddenly jet a bunch out um as the plunger comes loose. So right, we, you can either pretend that I'm working in a still air box or you can pretend that I'm working in front of clean air. So we're in front of our flow unit here. I'm going to take the lid off my spore syringe and I'm going to lift. What I like to do is is effectively clamshell, which I'm going to show you from the side. Clamshell is like as if I'm opening up like a little clamshell. I open it up into flow so the clean air is heading towards me. Oops, don't do that. And then I'm going to take a tiny, tiny amount of spores and I'm just going to drip, drip, drip. Three drips on there. It does not have to be three drips. And yes, I did just push my hand into the scalpel blade. Um, don't do that. So you got your a couple of drips on there, and then we're just going to roll it around the plate a little bit to coat the surface. And that's it basically. We've got the spores are now on agar, so they've got everything they need now to germinate. So what we would normally do at this point, I'm going to today use a bit of grafting tape. So you can use parafilm or grafting tape. So grafting tape is basically like this cheap stuff you can get off like from garden centers uh to I think they use it to like graft twigs and stuff for trees. But as you pull it off, a lot of things that are created like rolls of tape. They are actually sterile coming out of the factory. So but this part of the process is really important. At the moment, this plate has there's airflow that can travel through the sides of here. These are vented plates. So, airflow can travel in and out of this plate if it's not sealed up properly. So we're gonna see it. Do this well on where you can actually see it. I'm gonna run this around the outside, making sure to kind of seal up as I go. Doing this with gloves on is actually a nightmare. All right. So I've gone all the way around and I've sealed it. Pull that off. And now you've got effectively this sealed plate that's sealed all the way around. This can now just be put into incubation. So um, all the incubation sort of stuff we're going to talk about today is going to be around between 30 degrees, sorry, between 30 degrees, for Christ's sake, don't do that. From about 20 to 25 degrees. Things will germinate at lower temperature and um, but things do take longer. Like mycelium tends to slow down a lot if the temperature isn't correct. So you will see germination, but the whole process is likely to take a lot slower if you don't have the right temperatures for incubation. So all of the incubation we're going to do tonight is in between 20 and 25. I'm going to move this scalpel out of the way because I think I may have actually nicked myself underneath the glove, but we'll worry about that later. Um, when you're doing this, you um, typically you create sort of seven or eight, 7 or 8 of these plates and just have like a bunch of them there all colonizing because some of them are going to be contaminated. Some of them are, some of them are going to be good. What we're looking for when that mycelium starts to grow out is we're looking for just a clean piece of mycelium that starts to look like it's a little bit thicker. So we talked about rhizomorphic mycelium. This is more, when you're working with spore syringes, typically people are talking about, mushrooms that produce this sort of mycelium. So it's a good way uh, of just identifying mycelium that is definitely, is going to work out for you later on in your grow. I need to wonder if there's actually blood underneath that glove there. Like when you think, oh, I'm actually bleeding underneath somehow, cut myself underneath my glove. Brilliant. Um, so we've inoculated spores onto our agar, and now we're going to wait for that mycelium to grow out. Normally, this part of the process, can take sort of up to 7 days. It Patience is the key with a lot of mycology stuff. If the conditions are absolutely ideal and everything goes really well, and the spores are really viable, and in a best case scenario, you might start to see some growth after 3 days. If you start to see growth like the next day, that might be a bit of a warning sign there. So if you start to see very fast growth across the plate that looks really fluffy, like within like a day or two, normally that'll be some sort of mold that's actually that's growing out on the plate. Um, so there are certain things that we can isolate quite easily. Um, Someone did ask actually about incubation. So I used to do stuff just in my airing cupboard. You can also do we sell an incubator that's basically like a bain marie style thing. You don't have to buy the incubator, by the way, but what it is effectively a fish tank heater with a small amount of water in the bottom of a plastic tub, and then another plastic tub inside that, and it sits on the warm water and creates like a nice little tub for you to just put all of your mycology stuff in. So you can make one of those yourself. They're pretty easy to make. That's a really good way of doing stuff during the colder months. Obviously, during the summer, when heat isn't really an issue, you can just grow this stuff. This will just grow out in your normal somewhere in your house that's a evenish temperature. Um, so let me just see what if there are any questions just quickly above that relate to that. Do, 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 do. So yeah, we talked about heat pads. So heat pad, the issue with using a heat pad is that the heat's a bit too direct. What can happen is it doesn't create an ambient heat temperature. It tends to just dry out agar and dry out the bottom of grain bags. So when we move on to grain inoculations, if you put grain directly on top of a heat mat, it will all the grains on the bottom of the bag will dry so much that the mycelium won't bother with them as a food source. So it can be really, it can really scupper your efforts. Um, right. So we've basically I'm going to I'm going to take an example of a plate that's looking really dog shit that I've got. So here's a really good example of bacterial contamination. Um, this is a wild oyster from just around the corner from um, the Forest of Dean. So we're trying to grow out a lot of native mushrooms at the moment. So this is a piece of tissue from a relatively dirty mushroom. Um, so Simon, what would Simon Rowe asked, would this process differ if using spores as opposed to LC? So typically, once you've got a sterile liquid culture, you wouldn't need to use agar as much because you can just continue to expand out the liquid culture. The process I'm taking you guys through right now is to actually be able to produce liquid culture. So first of all, we're effectively cleaning up the mycelium on agar, and then creating a sterile foundation to transfer to liquid culture. So here's a really bad looking plate. Um, a little hint as well, like I talked about us using like a professional flow hood. We would not open a plate like this in front of a flow hood. Any contamination that we work with, we would still tend to work in front of in a still air box. What we're obviously trying to avoid is just blowing contamination out into our clean space. So when you have something that might be, that might cause issues in the air, we tend not to want to blow it all over the place. I'm going to hold this up to the camera a little bit. So we're talking about the food coloring as well, like the mycelium where it's like it's having troubles with bacteria. It's like actually drawing the food coloring out of the liquid, and it's producing these really weird metabolites that are like a reaction to the bacteria. So one thing that's not really acceptable to if you're buying a spore syringe and you get loads of bacteria, like this kind of milky substance that is produced whenever when instead of seeing mycelium germinate, you end up with like a milky liquid on top of your agar. Um, that is generally seen as something that is a good argument for getting a replacement sent out. Bacteria is a really difficult thing for us to deal with, and that tends to come from the process, the actual process of it means that the batch was contaminated. So it's a fair thing then, I think, to reach out to a vendor and say, look, I have bacteria, in this, and it's not something I'm going to be able to easily deal with. Um, so right, I'm going to show you how to basically transfer away some of this. This is a really grotty looking plate. So let's say, we've germinated our spores, and they're looking like this. We've got some viable mycelium on there. Arguably, like if you've got better looking plates than this, you would you would not go for this one, obviously. So I'm going to take another plate off the top. Right. Um, yeah, if I'm working with like this sort of bacteria, like the problem with bacteria as well is that like often it almost like piggybacks with the mycelium. So you can end up with, even when you take a piece of clean mycelium from this, you can end up with a with mycelium that just isn't that great. There are a bunch of sort of more advanced techniques you can do to transfer away from bacterial contamination, but let's for argument's sake say that this isn't bacteria, and actually that like this area here and this area here is just like that there's some mold or something. Um, so what we would do in front of our flow unit or in our still air box, we would look for the cleanest area of mycelium that we've got in here, and really that in this instance is around here. We're going to take our scalpel and we're going to carefully cut a little square like this. And if you want to do a couple, say you're doing like 3 or 4 plates, um, you can just do a couple of little notches like this. So we've cut that out right the way to the bottom, so these should come out quite easily. And then what we would do is just with the tip of our scalpel, and just so if you're buying a scalpel, don't bother buying the sort of round-ended ones, because they're really not very practical for mycology. These are, the ones with the little the really sharp end. They're good at stabbing yourself during presentations, and they're also really good for like picking out little awkward pieces of mycelium like this. So we should be able to just lever this out like this, pop our plate down, and then using the same clamshell technique from before, we're just going to pop that on the middle of the plate, close it up, seal it up, and then a really important step, when you're finished, make sure you label your plates. So this is a mad looking lion's mane from Andy Knott, who is from Jurassic Coast mushrooms. And yeah, you can see all the mad growth patterns. It's even got little mini lion's manes growing off the edge of it from where it's been sat for so long. Um, Make sure the information you want to write on these plates is firstly what's in it. So you've got a Somerset lion's mane, um, the date, so if something's just not progressing very quickly, you need to know when you inoculated it. And then other things, if you want, if there's other information you want to track, it's good to just put that on the plate. Um, you can yeah, once you've done all your plates and you've got your little stack of sealed plates, um, we've got one example here of your transfer. We've got one where we've germinated some spores. What I would hope now with this is that we would see some really clean mycelium start to grow out across the surface. And as long as there's no sort of warning signs, like there's no sort of like gloopy looking halo around the agar. So sometimes you'll get like these growth patterns where this is probably a good example actually, where the mycelium grows out and then you'll get almost like a shiny edge to the mycelium. This can be an indication that there's still bacteria in the mix. If that happens, normally it's good to just go back and try and get a clean culture from, just try and start again. If you're using spores, just go back to the beginning and try and get something that's not bacterial. If you do a transfer and there's like mold on there, then you just repeat the process. So, you would wait for some good mycelium to grow out, and then you would transfer again to the next set of plates. And sometimes this can take 3 or 4 stages and 3 or 4 transfers to get a finally clean culture. Um, one more thing I'm going to show you quickly is I'm going to show you cloning. So we talked about different ways to introduce mycelium onto agar. So we've got our plates germination. Just put a lid on this horrible looking thing. So we talked about cloning. This again is like a subject all unto itself. Um, you can take a small amount of the of mushroom from an actual mushroom itself, and you can put that directly onto agar and grow out mycelium as if it was being transferred from another plate. This is quite cool because you can go to like a supermarket and take a mushroom like this. You can break it in half, which I'm going to do now. So you can break it in half. And when you do this, it exposes this area of flesh here. So this is all sterile here. Um, and the reason it's sterile is because it's been closed off to the air, and it's that mycelium will naturally form a barrier against other types of contamination. So what you can basically do is snap your mushroom in half. You can take a small amount of mycelium from here in almost the same way as when you take a transfer from agar. You can take a small piece of that mycelium and put it straight on a plate, and it will grow out exactly the same as doing a transfer from mycelium to mycelium. This part of the mushroom, it is just mycelium. It's just a different configuration of mycelium. So, this is a really cool thing to practice. One of the interesting things about cloning versus cloning is that you will take on the traits of this mushroom. So if you've got like a mushroom, let's say you're doing a grow and you end up with a mushroom that is white instead of brown. You end up with a single mushroom that's like bright white, really interesting looking. You can clone a small amount of tissue from that mushroom, put it on agar, take it through the process of through to liquid culture, and you can create an entire flush quite often of completely white mushrooms. So yeah, it's cool. It's like it's good to be able to do these kind of like genetics experiments and actually just do selective breeding with mushrooms just by um, as you grow over time, you just end up seeing unusual looking mushrooms, and you can almost just steal that look and save it for later use. And it's when you see unusual phenotypes. Ooh, wait there. Let me know when it's back. I need to find next time. I think I need to. Can you hear me again? Good. Um, when did I cut out? You don't know, I guess, because you weren't here. Um, so I was just talking about being able to take the genetics from the mushroom if you're growing out a flush of mushrooms. Stuck. What a nightmare. I think in future I'm gonna need to find a way of having myself directly plugged into the internet. Maybe it's wireless that's causing the issue. Can you hear me again? Hello? Hello? You can't hear me. Is it because I'm not speaking? It keeps freezing. Let me just I'm going to lower my video quality. Let me see if I can lower my. Let's do this. I'm going to take it off HD video, and then hopefully that will help. Um, Let me know if you can hear me better now. I'm going to keep moving my hands. Getting better. There you go. Okay, cool. Yeah, maybe that like it's gone here. Yep. Okay, we're looking good. We're looking good. That may be a good thing then. All better. Right, cool. I've lowered the resolution of the video, so it's not trying to stream a high resolution video, I guess. Hopefully that fixes the issue. What I was just saying was, did it break out? But did you see me doing the mushroom cloning? Did you see me cutting the bits out of the mushroom at least? Yes. Okay, cool. All right. Well, that's good. So what I was just saying is, if you're growing out, things like a mushroom, quick adapter. Thank you. Yeah, yeah. No, I worked in tech from the age of 16 to the age of 30, 35. So yeah, I do tend to think about quick technical things. Um, so you've grown out like this beautiful flush of mushrooms in a monotub or in a grow bag, and you've spotted this beautiful white mushroom. So I spoke about the white mushroom. You guys probably already heard about this. This cloning process allows you to continue the genetics of that tissue, and potentially have flushes of beautiful white mushrooms moving forward. Um, some of the other things also count as well. If you get a particularly large like mushroom, something that just really looks remarkable or has like some weird like quirky maybe the cap is looks different from some other mushrooms that you've grown, or all the rest of the ones in the flush. you could potentially continue those traits as well. So, these are what are known as phenotypes. So they're like expressions of genetics that just come up sometimes when you're growing mushrooms, and they do tend to come up a bit more when you're growing from spore. And the reason being is, when you inject a bunch of spores into grain, you're getting all these different genetic possibilities growing, out from each hyphae is like it's like a separate child. Whereas the cloning process is the cloning process is effectively just taking that, the one kid and just copying him. So yeah, you do sometimes end up with more interesting genetic diversity when just chucking a bunch of spores into a grain bag. But you do run the risk of introducing contamination and that being the thing that outpaces things. Right, I'm dragging on a bit here. We're almost at like God, we're at 8 o'clock. Um, so we've basically got to this point now where we're growing out mycelium. We've cleaned up a culture. Um, what we would normally do then is we have liquid culture here. So I've this is basically liquid culture is the same nutrients that were in the agar, but without the gelling agent. So this has got a very small amount of malt extract in distilled water. It can be tap water if you're doing it at home. We then sterilize this, and then we're going to introduce mycelium into this broth. The mycelium is then going to grow through that liquid, and you end up with something that looks like this. So this may be a difficult thing to see on camera, but there is mycelium growing through that syringe. So the liquid will end up looking like it's full of mycelium growing in all different directions. And the cool thing is obviously we have removed any contamination through a process of isolation on agar. We've now got this like perfect clean culture, which I'm going to use this one as an example. And we are going to transfer that into our sterile broth. So in the same way that when we did our little notch earlier, we're going to just take a really small wedge, and I'm going to talk to you. I'm not going to go into too much detail about like where is the best place to select from. If this hadn't grown all the way, this is an older plate, so it's grown all the way to the edge of the plate. Normally, you want to do your transfer when it reaches about 80% of the way. So, like, let's say use that as a boundary there. You want this kind of leading edge of mycelium. Um, you want to kind of be taken from there. I'm just going to go around there. So imagine the mycelium is growing out to that boundary and there's still a clean agar all the way around the outside. That's generally when you want to do your transfer because there's a leading edge of mycelium there that has a lot more energy, it has a lot more vitality than the rest of the organism, because it's the bit that's responsible for searching for nutrients. So if we are taking a bit of this mycelium here right on that leading edge of the rhizomorphic growth, we know that this bit here is going to be really searching for food because you're almost like taking the mouth of the organism and transferring that to liquid culture. Um, could you clone cordyceps? I'll answer that at the end because it's a bit cordyceps is a rule into itself. So I've got my little piece of mycelium here. We're going to transfer that. I really want to make sure I'm not going to stab myself again today. Um, so again, in a still air box, you would wipe this all down. You'd spray it down with isopropyl, make sure everything's all really clean when you're working in a still air box. We take that small piece of mycelium and we would drop it into the jar. You don't want to use a big old wedge of mycelium in here, because so um, we use something at MycoPunks called and a lot of cultivators use them called a magnetic stirring machine or a magnetic stirrer. So inside this jar here, I can't tilt it sideways very easily. There is a little white bar which you could probably see. That's a little magnetic bar. We can put this on a machine and it will spin that bar inside the liquid. The bar's been sterilized along with the liquid, and it will break up the mycelium each day. So as the mycelium starts to grow through the liquid, you can effectively put it on one of these magnetic stirrers and it will break apart all the mycelium, and then all that mycelium will almost have more jumping off points into the liquid. So it can really speed up the colonization of your liquid culture. Um, Normally this process here we've this could take a couple of weeks to get to a clean culture from spores. Now once we transfer to liquid culture, this can take normally 1 to 2 weeks to create a liquid culture. But once you've got it, this is only a tiny little jar. You could do this in like a liter jar, and then you've basically got a clean culture that you know is good. You know that it's going to cause you, 99% less issues than spores, and you can just keep drawing from that liquid. And you don't have to keep going back to agar once you get to a certain point. Once you get down to the last, say, 10 milliliters of liquid, you can just draw that up with a syringe and inject it into the next pot, and it will continue the process without having to do all the agar stuff again. Um, Lastly, I kind of wanted to just talk quickly about what we tend to use agar plates for is once we get to that point, we might want to, we don't want to lose these genetics now. We've done all the hard work. We don't want to have to like go back to spore and potentially lose our progress here. So I see this as being a kind of like a save point in a game. You've created a clean culture. You really want to kind of have, you might want to create sort of 5 or 6 plates. Take them to that 80% mark, and then I would stack them up in a clean bag or wrap them in cling film, and then I store them upside down in the fridge once they've colonized to that period. The reason you stack them upside down is you don't want loads of fridges generally. The temperature is fluctuating, so you end up with loads of condensation coming out of the agar. And you don't want liquid just sitting on the surface of the agar. It will affect the mycelium growth. So you can keep that in the fridge for sort of like a good 3 months before you need to then take it out and do another transfer just to keep things fresh. I said earlier about the temperature will slow down mycelium growth. That's kind of what we're doing in the fridge. But yeah, you want to make sure that the plates are just stored upside down. So Steve asked, how many times can you move the last bit of LC onto the next LC jar? So there's a couple of different thoughts about this. There's, the concern that we have, like as commercial cultivators, is something called senescence, which is where the the mycelium basically gets tired of reproducing cells. It effectively will slow down, and you'll end up with a yield drop off. That normally takes quite a while to happen. So like if the mycelium is given a slight adjustment in nutrients between different jars, often that can maintain vitality for like for just dozens of transfers. But it's also why we keep those, we keep the backup plates. Um, if you do notice, that you're starting to get less yields over time and you've been transferring and, doing a liquid culture to liquid culture to liquid culture, then you return back and we start back at the parent plates. So we would go back to those first generation plates. We would make sure things are right on agar again. We'd do a couple of transfers just to make sure that the growth is still all good. And once we're happy with agar again, we'd go back into liquid culture. So Joey asked, if you introduced mycelium to another mycelium, would they fight? So depend if they're the same species, occasionally, so right at the beginning of the right at the beginning of the video, we looked at like the mushroom life cycle. So, even when two spores are from the same species, they have to be the right mating pair to create mycelium. So when if you have like 10 spores, you'll end up seeing, let's say and I'm reducing this obviously drastically, there would be thousands of spores on the plate. Once the stuff starts to germinate out, you will start to see these natural barriers forming. They're called zones of inhibition, which is like a biology term for effectively like a no man's land of where two organisms meet and nothing will grow in between. So if they're not compatible, these two things, you'll see like this light mycelium move right up to the other mycelium, and it will just not connect at all. It will create this little barrier. So yeah, they're generally quite cautious of each other. Certain species within the same genus, so like you might have there are some oyster mushrooms that are not the same species that you can still mate together, which is how some of the more interesting strains of oysters have been created, by actually mating stuff that's not even the same species somehow. Um, but yeah, they don't fight. What you will sometimes get is if you're dealing with competing contamination like trichoderma or which is like the blue green mold. That can sometimes be a bit of a battle where mycelium will could just completely consume the trichoderma, or the other way around. If the trichoderma is like strong as hell, it will start to just eat the mycelium. So um, it's always an interesting thing. This is when you start to work with agar, you'll get to see this play out in real time, and you can really see you can kind of really see how things work. Um, you know, um, do you know I am going to very quickly cover the easiest fuck agar pots because I have got some here, but I've just realised how much how much time I'm taking, and I know it's Friday night, so I know that some of you guys you probably want to jump on the PS5 or you know smoke some weed or whatever it is that cool guys do these days. Um, so we created these little things called easiest fuck pots. This is a way of you doing stuff without having a still air box. We're here for the long run. Yeah, we'll see when the numbers drop off at off at 9. So we created these little pots called easiest fuck pots. You may recognize these pots from if you are a takeaway aficionado like me. Yeah. What we've effectively taken is what are called Satco pots, which are the ketchup pots. These are sterilizable because they're made out of polypropylene. We drill these little holes in them and put these ports on, and then we put some agar on the bottom. Um, yeah, I know. I'm PS5 is well. PS5 is going to get some action over the weekend, but not tonight. I think I'm going to be done in. Um, so the way these work is effectively they allow you to germinate spores without the need for a still air box. And what we effectively do is I'm going to just really quickly. I have done a video on this that's much more in depth. Um, So what I wanted to do, one of my things was is I got really fed up of people just using spores directly into grain and not having, because they no not many people have got time to create sterile environments or buy a flow unit or whatever. So I wanted to create this like workflow that let people work with agar, germinate spores, and then get that into liquid culture, but without the need for a still air box. How's the video quality now, by the way? Now, is it all good still? Sound's not dropping off? A lot better. Brilliant. That's great. Um, so what this is it has like a little injection port here and it has a filter that allows airflow in and out. Um, Imagine this is a spore syringe where earlier on we introduced just a couple of drips of spore solution into our onto our agar dish. We're effectively doing exactly the same thing but into a sealed container. Um, I'm skipping steps here. Like this should have been wiped down. There are all sorts of steps I'm missing, but there is a really good video on our really good. It's an okay video on our YouTube channel, which is just youtube.com/MycoPunks. Full guide on how to use these, because there is it's a cool process. It lets you work with agar even if you can't be bothered to do a still air box. So we're effectively just introducing a couple of drops of spore solution. We would then we'd incubate this exactly the same as a petri dish, let the mycelium grow across it. If it all looks good and you're getting good growth in the pot, you're kind of like, you can rest assured that you're kind of okay there and that like the risk of contamination moving forward is good. So once you've got that mycelium growing on your easy as fuck pot, we would draw up some of the sterile liquid culture out of our pot. So this is, now imagine this doesn't have mycelium in it, and this is just the nutrient broth. We would draw up a full syringe of that, and then we would introduce the liquid culture media to the agar instead of the other way around. So we'd slowly flood the dish. And what you will notice is effectively what we're doing is we're creating like a miniature liquid culture jar, but just in a ketchup container. So in here is like now flooded with liquid. And what you'll see is you'll start to see the mycelium climbing off the agar in the pot and into that liquid culture. So normally you'd incubate that again for a couple of days until you start to really see like the mycelium climbing through the liquid. And then you do everything in reverse. You take this would be a clean syringe each time. You would draw up the liquid with the mycelium in it, and then we would put it back into the main the parent liquid culture pot. And that's a way of you working. That is a way of you working with agar without a still air box, and without any kind of over the top, like say over the top, without using sterile technique. It's a really cool thing to try. These are quite cheap as well. Like, we do, I think they're like 8 quid for like four. So you can just have a go at this and like mess it up and not feel you you can just not feel like you're wasting too much money or going too far down a process. But yeah, it's a cool solution and can I draw it back up and use it directly into a grain bag? Yeah, exactly that. Yeah. So you could, if you wanted to go directly from this, draw up the liquid culture, and then put it straight into a grain bag, then you're good to go as well. It's exactly the same as taking it out of this pot. What this does here, this expansion of mycelium just lets you have like a big jar of mycelium that you can use when you want to. And once this looks like it's full of mycelium, you can just pop that in the fridge and it's good for like 6 months. Um, so yeah, it's a really good way of just having um, just a stock of liquid culture for you to use, and that way you're not having to go back to spores or you're not having to deal with agar all the time. Um, right, so I think we're kind of like I want to just check my list just to make sure that we've covered everything in that section. God, we're so far. I guess we've covered two sections. We've covered working clean. Um, so yeah, in terms of like vector for contamination. Um, there are a bunch of different does injecting more liquid culture equal faster colonization? Yes, it does actually. So the difference being, right, so one thing that doesn't, if you inject an entire spore syringe full of spore solution into a bag, what it does is it creates a potential anaerobic environment where there's too much water in the bag, and that can create like a stagnant water which will scupper your grow completely. So normally when you're working with spore solution, if you have to do it straight into a grain bag, then you would just do like 2 milliliters. These bags are hydrated. The bags that we sell, which I'm gonna bring over now because we're moving on to this step. Um, so these bags, the grain has been put to a specific hydration level. There's no standing water in these bags. You will notice some contamination. Yeah. Ugh. You will notice some condensation. Here's two different types of bags. Let me move all this shit out of the way quickly. Pop the lid back on this. Right, that can go there. Mushroom can go out the way. Let's get all of this stuff out the way. Do, 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 do. Right. Okay. Moving on to the next bit. So yeah, in terms of like let me just go back to that question. Sorry, one sec. Do, do, do. So you were saying about I was saying about so injecting more liquid culture will get faster results because the mycelium. I don't actually honestly know why liquid culture doesn't create an anaerobic environment. But you could like put a whole spore syringe full of a whole liquid culture syringe into one of these bags, and the mycelium is like happy living in that broth as well as growing through the grain itself. But if you did it with a spore syringe, it will just creates like a bacterial environment where you'll end up with just like a big patch of gooey mess at the bottom of your bag. Um, I'm just going to answer a couple of these questions really quickly. Mine are incubated at the moment, very fluffy, uniform growth. That's good. Is that to test your culture? Yeah, so the little pots can be used if you have liquid culture that you haven't used for a while and it's been sat in your fridge, and you don't want to waste your time, kind of moving on to grain and doing a bunch of grain and then realizing that the liquid culture you're using is shit, you can just inject a small amount into these pots, and they're great for just quality testing as you go along. In the same way that like, if you want to test a spore syringe for any bacterial issues, you can do exactly the same thing. But yeah, in terms of like the vectors for contamination, you know, obviously human beings, um, if you're not wearing gloves, and you're not sort of spraying things down, spraying yourself down, or if you're wearing like dirty clothes, or if you haven't had a shower in a week, um, these are all like vectors for contamination. Um, yeah, there's basically like that's the sort of main one, I guess, is human beings. Spores obviously spores to straight to grain is a vector of contamination because spores aren't a sterile product. Another thing is I said earlier about airflow. So there's two sides to that. There's too much airflow, which is blowing contamination in from around the room, and then there's not enough airflow, which is an issue with grain as well. So if I had to like choose the thing after spores that causes the most contamination with customer grows, it is people leaving the bag in their grain bag in the original packaging. So obviously, when your bag is in the box, it's all wrapped up, ready to be taken out, or it might be in one of the express pack bags that we have. Um, this is a bag that's hydrated grain, so as it goes through temperature fluctuations, it starts to produce condensation. Um, this white filter patch here, this allows airflow in and out of the bag to because mycelium breathes in oxygen and breathes out carbon dioxide, so it still needs to be able to breathe. This filter effectively blocks contamination from going in and out. If condensation builds up on the filter of this patch, mycelium will push its way, mold mycelium will push its way through that filter and it will bridge down into the bag. So it's actually able to bridge through the filter in this on these bags and get into the mycelium. So in the same way that like if you leave it in the packaging, in the original packaging, it creates just this environment where condensation is building up on that filter patch and it's got nowhere to go. It's like the inside of your like your washing machine, the seal on your washing machine when you keep the door closed, it has that that high humidity, and it's just a breeding ground for bacteria and other problematic things. And the mycelium just will force its way through the filter into the bag. So the amount of times when someone so we might get someone who says like, "Oh yeah, I've got contamination," and I look at like when their order was, and it's like 6 months ago, and they've remembered that they bought a grow kit from us back in like July, and they've just taken it out of the box and all the grains all ruined. That is a really surprisingly common one where people just leave it in a high humidity environment. So when you do receive sterile products from us, just make sure that like they're taken out of the packaging, that the airflow is allowed to get to these filters, just because that way, it keeps the humidity off them and prevents contamination. Right. You cannot mix traits from two dicaryons on a plate. From two dicaryons. Um, no, I think you're right, actually. Yeah, I don't. So I've not done that much breeding. So the person like a really interesting person to speak to about breeding would be there's a guy called so I don't do that stuff very much. Um, There's a guy called Andre from Fat Fox Mushrooms who did a really good talk this year at All Things Fungi Festival. And he did a full talk on breeding and monocaryons and dicaryons. For anyone who's like wondering what that is, so a monocaryon is basically like the pre-mated hyphae thread that's it's just one mating type, and a dicaryon is where the two things have met, and then they become dikaryotic. So I think I got that the right way around. So yeah, yikes! Better unpack my parcel ASAP. Yeah, I don't. I'm making sure I keep an eye out for someone called Dave now in tickets. Sorry. So Andrew was from Fat Fox Mushrooms. They're like they are a mycology company in the UK that do like all sorts of cool grow kits and stuff like that. But his talk was like super interesting. So, all right, quick sip. It's good to see that there's actually still 30 people in here. Like, I'm really impressed. And I'm really happy that you're sticking with me as well. Because I was like, it'll start getting to that point. And I was keeping an eye on the number because if I start to waffle and that number starts to drop off, I'll just change the subject. So it's a good indicator. Right, let's talk about grain spawn. Um, we're going to do a Q&A at the end. We're going to do like a more generalized Q&A at the end, and then people can, just taper off if they don't have questions. Firstly, what's the point of grain spawn? So, we're basically looking for a way to create a lot of mycelium without it fruiting. Um, that's basically the best way of putting it. So, we want to be able to create a lot of mycelium that is like almost ready to fruit, and then we want to be able to expand that out further into substrate and have like more of a like an overall mass of mycelium that will give a big surface area for mushrooms to grow from. So these bags. The way they work is the filter on these, they restrict airflow. So if you will imagine, as mycelium is growing through this, it doesn't get any changes in air pressure. It doesn't, as far as this is concerned, as the mycelium grows through this, it's as if it's within some wood, in nature. So it's seeing no changes in air pressure. It's got an abundant food source. So there's nothing that makes the mycelium feel like it's threatened or that it should be suddenly, um, should I be releasing creating fruiting bodies because I'm losing my food source, or are the conditions becoming so unfavorable that that I need to basically pass on my genetics. Um, I mean, what you may notice sometimes as well. So this is a good example of like you may be growing doing your grain spawn. You may notice that there might be some mushrooms growing in your grain spawn. What this can sometimes be is like it can be where mycelium has effectively come up against some stress in the bag. It might be like a little bit of bacteria that has gone unnoticed. It might be some, piece of contamination that isn't visible, but or it might be the conditions in the bag have changed. So maybe the grain is getting a bit dry over time. Um, or it could just be like a drop in temperature. Once mycelium feels threatened, it does what like human beings do in stressful situations. It starts shagging. So it produces it produces fruiting bodies. This is why you get a lot of children born during wars, right? Um, it will produce fruiting bodies. It will produce the next generation of its offspring because it feels threatened. So the grain spawn stage of mycelium production is effectively about making mycelium feel as safe as possible and well fed, well taken care of, but also we keep the airflow low because we want to to kind of make it feel like it's below the surface of the substrate and that the conditions aren't right for fruiting yet. Um, what this allows us to do, as a gourmet cultivator, it allows us to very quickly ramp up the amount of fruiting blocks that we can make. A small amount, you only need a very small amount of grain spawn in a rather large substrate bag to get it to fully colonize. Um, and what it lets us do is it just lets us make, produce mycelium at a really quick pace. And mycelium grows through grain much quicker than it would through substrate. So if we just introduce mycelium directly to substrate, it would really struggle. It would be like, this is not enough nutrient for me. This is not something that's conducive to me feeling happy. So you're more likely to get sort of issues of just slow pace. What this is effectively doing is we're going to create a big mass of mycelium, which is going to look like this. So this is going to look a bit weird, this mycelium, by the way. This is Foliota adiposa, I think, which I know someone's put FA on there. Foliota's with a P, but so this is like the chestnut mushroom. So this is like got like ginger mycelium. It's like a bright orange mushroom that has like a ginger mycelium to it. So this you can see mycelium's grown all throughout the bag. Um, the process normally would be, we would inoculate this with some liquid culture, or we might drop some people might drop a piece of agar directly into a bag in front of the flow unit. We'd allow that mycelium to grow throughout the bag. And once we see like a quite a decent sized patch of mycelium, um, what we would typically do, once it's about 20% colonized, we would break everything up. Um, and then we would redistribute all the sort of myceliated grain around the bag, and then you would see this sudden acceleration as the mycelium jumps off all the new places in the bag. So normally, like the last part after what we refer to as a break and shake, that part is generally really quick. You've got this small amount of mycelium that is now being has all these different jump off points to fresh food around the bag. So like it just speed things up ridiculously. And it's part of like if you ever buy one of our beginner grow kits, there's this is part of that process that once you reach 20% colonization, we do a break and shake and it redistributes everything in the bag. So basically we've got our grain. I'm going to grab my syringe back. Um, so so if you've ever bought one of our grow kits, basically comes with a grain bag like this. You come get a little injection wipe. We're gonna wipe the port on here, allow that to dry. This is an empty syringe now, so I'm gonna have to pretend like there's mycelium in here. This feels like blasphemy putting just a bunch of air in a grain bag, knowing that I'm just ruining it. Um, so we're basically going to put the syringe straight through into the bag. If you are using spores, we're only going to use 2 milliliters, so you just put 2 milliliters in. If you're using liquid culture, you could use 5 milliliters. You don't want to be like, gratuitous with it unless you're in a rush. If you wanted to like really speed things up, you could use the whole syringe. If you're trying to make up for stuff that's been lost to contamination, maybe, or if you've had like, or if you're just in a real rush, then you can put more mycelium in there. And once that's been inoculated, ooh, fucking hell, that was close. That's what happens when you're looking at the screen, looking at the screen whilst doing one of the things. Um, Once you've inoculated that, you're going to leave that in a warm environment, 20 to 25 degrees, same as all the other bits. Mycelium's going to start to grow in there. If you've used spores, obviously the spores are going to need to germinate, so you're adding on additional time. Um, Once you start to see a little patch of mycelium, you know you're good to go. Um, Again, once that mycelium's grown out to 20%, you're going to really crumble everything up. And you can't damage mycelium like through pressure. So like you can really break this stuff up. And I'm actually going to just show you like when it looks like a solid block like this, it crumbles up nicely. Um, we're basically going to get all those grains. Um, And in the same way that I said about giving more jump off points for mycelium, when we add the better that this is broken up, when we add it to our substrate bag, it's going to have more jumping off points and it's going to colonize more evenly and more quickly. So yeah, what you don't want is like just big clumps of of grain spawn that are just sat in your substrate. So the better this is done at this point, the better the better the end process. Um, so Steve asked, what's the ratio of spawn to substrate with gourmet mushrooms? So this is sometimes like a factor of time. So if you're, it's normally way less. That if you were growing, like our Austrian counterparts, if you're growing magic mushrooms, we would typically use a lot more grain spawn in a lot less nutrient a substrate. So I'll show you the substrate in a minute, but we with gourmets normally about 20% of your grain, sorry, 20% of the weight of your substrate you would want to put as grain spawn. So I'm not going to try and do maths on a Friday night because it's after hours. Um, but yeah, you can put less in, but it takes longer. You can put slightly more in if you want things to go quicker. But you should just be aware that like when you're adding grain in, this adds to the overall nutrient profile of the bag. So like if you've got like a low nutrient substrate, like a maybe like a wood substrate with bran in it for growing lion's mane, and you add lots of grain spawn in, you've got to account for the additional nutrient of the grain spawn itself. Um, One of the things to kind of note here is that this is a point where a lot of people impatience can cause real issues for you. If you if you open this bag without the grain being fully colonized, you are effectively inviting any other organisms to jump on the remaining grain, and that grain that will then start spreading through the end substrate. So be patient, let this fully colonize. Even if there's like a little tiny spot of grain in the bottom and it just doesn't seem to want to colonize, what you can do is you can effectively just like disturb the grain at the bottom, kind of like break or do like a little tiny mini break and shake just to get it to mix in with the rest of the substrate and then reset it back and give it a couple more days. Same thing with the top of the bag. So sometimes you'll get like a little layer of grain across the top layer that just refuses to colonize. So you might end up and what that normally is it's just that the grain doesn't have good enough contact with the rest of the bag. So what you can do in that situation, I like to kind of break up just the top layer of the like solid block. I like to just break that up a little bit to bring some of that colonized grain in with the uncolonized grain. I give it a little mix around like this, and then I give it a slight tamp down like this, just lightly. And what that does is just gives everything a good it just gives everything a good surface area and good contact, and it allows that mycelium to jump up into the uncolonized grains. A multiple break and shake's acceptable if the bag still hasn't fully colonized. Yeah, I think, yes, to an extent. I mean, it sometimes if if you're using older grain and it's drying out a bit, you may require multiple break and shakes just to just to get it to really jump off onto those dry grains. So yeah, it's not a big deal to continue to do that. Obviously you want to look out for warning signs of that there may be something else going on in the bag. If there's like any milky sort of like residue in the bottom, then the bag's bacterial. All of these things can lead you to to troubles with colonization that might just be, that may be the reason that the bag's not colonizing. So just yeah, just be aware of there being other things. Another a little tip with grain spawn is once you sort of become accustomed to growing mushrooms, you kind of get to know what the mycelium smells like. So oysters have a very specific smell. Um, you can smell through the patches on these bags. So it's like you're growing. I mean, cubensis has a specific smell. Oyster mushrooms have a specific like sweet smell. So you can literally just apply a bit of pressure to the bag and smell it. If it smells sour, then there's more than likely a bacterial issue in there. So if it smells like just too sour to be the species that you're used to working with, then don't risk putting it onto substrate. Just go back a step. Try to work out where any potential contamination is coming from. There's nothing worse than like, than your effort than continuing ahead half cocked when you've put so much effort into the full process. So we've got good quality grain spawn. Um, we this literally is like if you can get to this stage, wow, this is like 95% of the job. You know, this part here is where like all the issues come from. If you can create good quality grain spawn, and you can just reliably have good quality grain spawn, then almost all of your problems as a cultivator are like, just fade away. Um, So I'm going to talk to you really quickly about substrate, and then we're gonna rush on to the Q&A at the end, because things that have overrun loads, I just need to know next time to be a bit more reserved in what we're talking about. Or we could start doing some more specific stuff. We could like go into greater detail of stuff. Um, so this is a substrate that we sell. We sell about 30, 40 tons of this a year. It's CVG, which is um, this was popularized with magic mushroom growers, and the reason it's such a good substrate is it holds moisture really well. There's no nutrients in it except for a bit of cellulose. Um, and what it does is it basically provides structure for mycelium, and it doesn't really provide any nutrients at all. So the risk of contamination when transferring grain spawn to CVG is just there's very little risk um, of introducing any contamination as long as your grain spawn's good and you've been patient and you've made sure it's fully colonized. Adding to CVG after that is like almost foolproof. Um, that said, it's like it's not impossible to have issues. Um, this is something you can create at home as well. Like it doesn't someone's just asked what's the best pasteurizer bucket tech. Bucket tech doesn't work for me. Um, if bucket tech just really quickly, Koya is basically coconut fiber. You buy it in these solid bricks from garden centers. Um, if you're making your own Koya substrate, you can basically just put some in a bucket, pour some boiling hot water over it, and put the lid on, and it will hydrate. As long as you put the right amount of water in, it will hydrate nicely. That surface, the shock from the hot water should kill off any surface issues. So any like mold spores that are in there. Normally, that's quite a good way of doing things. If you're finding that you're struggling with that, I would check the type of coir that you're using because some of them actually are inoculated with trichoderma because it helps with growing, which is what coir is intended for. So some of them come pre-inoculated with coir. So not all coir is created equal, I guess, for mushroom growers. And then I would look more to your grain spawn than I would um, worrying about whether your bucket tech is working. As long as the hydration levels of your substrate, I think this actually needs to be a completely separate talk. I can do a whole talk on making substrate. I'm going to just finish this one off, and then I'll make some time to come back to pasteurization. Um, so CVG, this is basically has enough water in it to where it holds within the substrate without pooling. So when we're creating substrates as mushroom growers, there's something called field capacity, and field capacity normally is just like it is a substrate's ability to just hold water, without pooling. The way we normally would do this by eye is if you're mixing up some substrate, and you've added your water to it, you would take a handful of your substrate and you'd give it a light squeeze. Um, you should end up with a small stream of water coming out. If it's dripping when you're holding it and not applying any pressure, then there's too much water in your substrate. That will definitely pool. If you can hold a handful of that substrate without it dripping by itself, and then you have to give it like a gentle, a squeeze to get liquid out, then you know that that's at field capacity. So this has got a few other bits in it. Um, there's something in here called vermiculite, which is basically like a volcanic rock that is really porous and holds loads of liquid, so it helps with keeping the bag hydrated. The coir obviously is just the lowest possible nutrient food source. And there's also some gypsum in here as well. So, we add agricultural gypsum into our substrate. Um, as you're kind of going through the process of pasteurization, occasionally it can change the pH of the soil. So, what gypsum does is it basically keeps everything within a certain range of pH. It stops it from changing during the process. It's called a buffer, basically. But again, whole subject in itself. Um, so the reason we use this is because it's very much foolproof for a lot of people. It's our least problematic substrate that we have. It's easy to make. It's great for all types of mushroom grows. The downside to have it having less nutrient in it is that if you're growing gourmets on this, you're not going to get the same size flushes and the same you're not going to get the same yields overall during your grow as you would do for a substrate that has nutrients in it. So if for instance you're growing oysters and you're growing them on sawdust and bran, for instance, the subsequent flushes and the size of the flushes that you get initially, a lot of that energy is coming from the nutrients in the substrate. So you do get better flushes with nutrient-based substrates, but they're also prone to contamination. So you do need to have like a sterile technique that stops things from getting in the bag and jumping on that, on the bran part of your substrate. Um, so yeah, this is great for beginners. It's why we push it so heavily, and like, it's because it's a very foolproof way of people getting into mycology without having too many issues. Um, so the last part of the process is basically to add your grain spawn into your substrate. Um, so this is once we've made up this substrate, it goes in we have a huge pasteurizer at work, which is basically like a steam generating tub that will fit like hundreds and hundreds of these bags in, and they sit for like 24 hours in just boiling steam, and that will kill off 99% of the organisms that we worry about as cultivators. It doesn't, there's some bacteria and things like that do survive that, but they're generally not something that's going to cause us issues. Um, so basically, I haven't got. I can use my scalpel. One last chance to cut myself. Um, so we've got our grain spawn. This is where I dumped this right onto my computer. Um, scalpel, scalpel. I would have put it somewhere out of the way, safe as well. Jesus Christ. Right. Right, I'm going to just use a scalpel blade. Right. So we're going to basically open the top of this substrate. So all these bags they're like they have this seal across the top where we heat seal them with what's called an impulse sealer. It's like a hot strip that bonds the bags together. And you notice again a big filter patch on this. So like I said before, mycelium needs to breathe. It breathes in oxygen, breathes out carbon dioxide. So the amount of airflow that's required for the fruiting process is slightly different to what we need for grain spawn. So the filter changes slightly. This is a what we call a B type filter. So it blocks a different size of contamination, and it also allows just slightly more airflow than the other bags. This is going to be very dangerous. I've just done my manager's health and safety course last week as well, so this is This would not be part of that. All right. So bags open. This is something you can do out in open air because, like I said, the risk is with your brain spawn creation. That this is where all the problems happen. This is not where the problems happen. So this is something as long as you've got your windows closed, you can do this in open air, and it's very unlikely to cause you issues. So the bags you'll find sometimes they're almost like vacuum closed from where they've been under heat and pressure. I'm going to open the bag up. I might have to you can just move this up slightly so you guys can see a bit better. Up you go. Ooh, hello. Right, there we go. It's better angle. Um, oh, that smells good. All right, so we've got our grain spawn fully colonized, checked. Nothing that looks problematic in there. So we're just going to we're not going to fully open the bag of grain spawn. We're just going to cut one corner of it. So I'm basically just going to just make sure I put my finger on the wrong side. I'm going to cut it across like that and give myself like a little spout to work with. Um, I said before about the amount of grain spawn we'd use for a gourmet grow. You know, if this was a wood substrate with nutrients added to it, then we would only put like 20% of the weight of the substrate worth of grain spawn in there. So a bag like this that's 2 kilograms, we'd put, someone do the math, we'd put that much in. So we've crumbled up our grain spawn. I'm going to stand up for this bit. We'll launch all this into the thingy. Let me just move that over slightly so you can see a bit better. Come on, do your thing. Ooh, there we go. Right, top of the bag's open. Literally just gonna dump this grain spawn in. Oh, you made the hole bigger. Right. Okay. So we got 1 kilogram of grain. So when when I talked about our Austrian friends who grow magic mushrooms, it's a 2-to-1 ratio for anyone who's from Austria in the chat. 2-to-1 is twice as much substrate as grain spawn. What I'm going to do to redistribute this, we are going to fold this in. Oh, sorry, we're going to fold it down first. So we're going to go 1, 2, 3, 4. Then we're going to fold it in on itself like this. And that allows you to basically grab this grain spawn, the whole substrate bag, and it traps some air in there. And this is going to let us mix everything really well. So we're just going to put it in a really good shaker. It'd be amazing if this just exploded on the stream. That'd be absolutely. Right, homogenized. Um, if you bought a grow kit from us before, the beginner grow kit it comes with this little pack of stuff. So right, everything we're in the final stages now of the work that we need to do. So in our little pack in here, we've got some paper clips. What we're basically going to do is we're going to tamp this down a little bit to give good contact across the substrate. So whenever we're leaving, substrate or grain or anything like that, and we want to just make sure that it's not pressed down too hard so that the airflow doesn't is affected. It's just we want good contact. So, we do want to create this good surface conditions as well for the mushrooms because this is basically like the last time we're going to touch this before it becomes goes into like fruiting mode. So surface of the substrate inside, we're going to just kind of like even it out. We're going to flatten it down so there's no like big fluffy bits sat on top. And you can do this through the bag. You obviously don't need to put your hands in. And what I like to do here is I like to push it out a little bit into the edges of the bag because what we don't want is like gaps where air gets down the side of the substrate bag. Just gonna move this down a little bit so you can see a bit better. It's not gonna let me. All right, better. All right, it's a bit better. So we're effectively creating the kind of sort of final surface part for your for before we start fruiting. So we're going to create that surface. Um, if if too much if there are too many gaps down the side of the bag, what you'll end up with is side fruiting. So you'll end up with mushrooms that are just growing down the side of the bag. Typically they're fine to, they're just as good as any other mushroom to eat. There's no issue with them, but you we kind of want to avoid that, that process. So what we do is we've got this big elastic band in with your in with the grow kit that goes over the top of everything. And we're going to create like a little like a basically like just a way of blocking the air from getting down the side of the bag because we want this side bit here to feel like like it's under the surface of the substrate still, and that way it should reduce the amount of mushrooms that pin on the side. Once you put your elastic band back on, kind of push things out again, don't overpressure it. That's pretty much it, like good to go then. So gonna fold down the top a couple of times just to make sure that there's not like, any big gaps where air's getting in. We're going to put our paper clips on. 1, 2, 3. Um, this is then going to go back into incubation until this bag turns completely white. So once the mycelium, you'll see all the mycelium after a day or two jumping off all the grains in here. You'll see them jumping off into the substrate, and what you should end up with is just this beautiful sort of white block of mycelium. Um, there's if you look at the instructions online for these grow kits, there's a troubleshooting part in there which shows you things that are red flags during the fruiting. If there's potential contamination in there, what that might look like. Um, so yeah, I would have a look at that because there is still a risk of that you didn't see something in your grain spawn, or there was like a bit of contamination in the middle of your grain spawn that went unnoticed. Um, Once this is turned completely white, we're going to bring it out of incubation into a lower temperature. So this can be like out in your front room or just somewhere where the temperature is, within the sort of like 15 to 20 degree range. And then for the first time, we're going to introduce a bit of fresh air. So once a day, you take your paper clips off, you open your bag up, give it a bit of air, and then put your paper clips back on. Um, and after a week or two, you'll start to notice the primordia bit from the graphic at the beginning. The primordia is where the small mushrooms are starting to grow. Um, and yeah, shortly after, you'll have mushrooms growing inside the bag. Um, all the humidity and everything should be taken care of with the funny, Susanna said, do you spray? So, the amount of liquid that is in the substrate and the hydration, the hydration levels, what that should do is basically keep the humidity exactly correct in the bag. So you shouldn't need to spray. If, for instance, like the surface conditions on of your substrate when it's colonized look, it looks too chalky, or if it just looks, if the whole bag is looking dry, then I would mist just the insides of the bag. So just try and mist the walls of the bag. Don't let water just pool onto the substrate, because that can just affect the mycelium growth. But yeah, you can just mist the inside of the edges of the bag, and then that should be good to go. But that's an unusual thing to have to do. Normally this method of growing is like the thing that makes it quite ingenious is the fact that it controls all the humidity within the bag. Um, and then you're good to go. Um, I think that's it. Um, there's loads of stuff I could have expanded on, and there's loads of bits that we skipped out on because we're not in an environment. We haven't followed the instructions to the letter here, is what I mean. Um, so I think that it's good to see the processes and kind of get some idea of the workflows and stuff. But obviously, just be aware that I've skipped a lot of steps just for brevity. But what I probably will do as a follow-up to this maybe is maybe I'll do some videos in our lab where we're actually creating some of these things in a lab environment doing proper technique. But these are all like the expansive subjects that we could really go into and take loads of time on. So I think you know depending on on how things look, I'd probably like to go into more detail on all these little niche things. and then you guys can pick and choose which ones you want to attend. I'm gonna make all of these free. You know, I'm not gonna I don't wanna I don't think this is ever gonna be something that we're gonna charge for. You know, I want you guys to feel more comfortable using our products. I want you to feel like you're learning something and that you're better informed to make good decisions about the products you buy. So it's you know, it's a win-win situation for us, and you know, and I you know, I want you guys to really enjoy this and you know, get the most out of it. But yeah, I'm going to open it up for Q&A. Um, we probably missed a bunch of stuff because you won't be surprised to know that being as ADHD as I am, my planning, I was still doing the PowerPoint presentation for this about 10 minutes before we started. So yeah, that what I want to do is I want to refine this, make it better, learn from the like little technical issues that we've had, and then you know create something a bit more like a bit better and a bit more engaging and a bit more interactive so you guys can, you know, maybe we'll do more polls and stuff as we go through. Um, but yeah, if anyone has any questions, I'm going to go back through. So Steve asked about, I probably missed loads of stuff here. I am sorry. Um, so less sub Russian so equals faster fruiting. So yeah, like um, with you can up the amount of if you're doing gourmets you can up the amount of grain spawn that's going into the bag just to get things to move on quicker. You know what we see with some of our commercial customers is that if like they have like some catastrophic loss, you know maybe they've like um overhydrated their substrate and that they've just ruined a whole batch. Often they will over grain spawn things just to kind of get things to catch up with production. Just going to switch my camera back so I can see myself. There you go. Um, yeah, so they will often, you know, just go heavy on grain spawn just to bring things up quickly. But it's not you know, grain spawn from a production standpoint is more difficult to create in bulk. So, you know, you people often, you know, as a commercial grower, they'd be creating a tiny bit of grain spawn each week and then loads and loads and loads of substrate because they only need such a small amount of grain spawn. So it's yeah, they typically will focus on. You can't see my face. Let me just put it on to share. Um Undo that. Content only. Oh, share. Uh oh, switch camera. There you go. Right. Um so yeah, yeah, like commercial growers will do think, you know, their process is going to be very different from a lot of home growers. You know, um yeah, I think even in terms of like the amount of effort that they put into creating really high quality stuff, like, you know, they're not going to worry so much about really high quality grain spawn because they're just doing stuff speed and efficiency is more important as a cultivator sometimes than like perfection. So you know, you'll see things very different. You know, when you visit a mushroom farm, you'd see things very differently to what you might do in you know, a business like ours where we're creating like quite exacting products. Um, you know, a mushroom farm is not going to be caring about clean grain spawn, like the actual inputs being clean. They're just going to chuck a bunch of stuff in the bag and sterilize it, which is good because there's actually, you know, it's much better. If you have two projects, sorry, I just missed one question above. Let me just see if I can move that out of the way. Participants, close that. There you go. It says you've flush. If so, Suzanne said if you're having more than one flush and the substrate starts drying out, do you soak the cake? I have six flushes in my last project. Um, so so with like subsequent flushes, yeah, you so there's a couple of different things you can do. You can either just continue to spray the inside of the bag to like to keep the hydration levels nice. So just misting the walls of the bag. You can soak the cake as well. So once you've done your harvest, you can. When we're talking about a cake, by the way, is we're talking about like a fully colonized block of mycelium that's in the fruiting stages. So you've done your harvest. You know, what I think is probably the best method is to completely submerge that block in water. So just put something weighted on top of it, submerge it overnight, or for 24 hours ideally. Ta Completely pat it down with a bit of kitchen roll. Make sure the surface is all dry again, and then put it straight back into fruiting conditions. That normally gives the best results. But you've obviously got to be mindful of the fact that like the mycelium is, it's probably eating a lot of that substrate, and you're more likely to get less and less as time goes on. Some of you might be familiar with like these absolutely massive mushrooms that you sometimes get on subsequent flushes. So like if you're on your like third flush and you might end up with like one massive mushroom. And going back to like that idea of like stresses in the environment, you know, the organism is basically putting all of its effort into creating one last good spore dispersal method. So when you get those big mushrooms, sometimes it's like the organism saying like, "This is it now. This is all I've got left." Uh, so yeah, I hope that answers the question. I probably didn't even answer the original question, but yeah, I did. Yeah, yeah. Would you use white rice as as grain spawn of blue oyster? You can use white rice. You can use packet rice. So if you look at Ben's Tech or Uncle Ben UB Tech, you can inoculate bags of rice that you buy in a supermarket. It can be a bit more difficult to track contamination with that, but it is cheap as hell. Like it's cheaper than the stuff that we sell. So it's definitely worth trying. I think if you're a newbie and you want to know the difference, then yeah, it can be a really interesting way of doing things. Um, if you have two projects on the go, can you store them together, e.g., in the same box through the same process, or do you need to keep them separated? So you can keep your projects together during like the colonization stage. Just be sure not to be blocking any of the filters on any of your bags. Think about the crowding and how it might affect the just the conditions and stuff and just make sure that there's no like you're not creating an environment for contamination like high humidity or anything like that. How did you come to mycology? You're obviously really knowledgeable, and I wondered if you had any formal. No, I don't have any formal training at all. I have ADHD, and it um it just ticks a lot of serotonin boxes for me. The learning aspect of it, the fact that like people like us can do, I think, real science in a home environment. Um, I think that that's one of the coolest things ever. And it's also an industry where citizen science and these sorts of projects are like really they're really respected by actual scientists as well, because we're gathering good data, and there's not much money in the science of mycology. So a lot of people doing the science are relying on people like us to do the experiments. But yeah, I just love it. It's very cool. But how did I come into it? Like so many mushroom growers, through a series of big psychedelic experiences that made me realize I had to drop everything I was doing in life and dedicate my life to mushrooms. Nice to see you, Dave. Yeah, thanks for joining. Is there any literature you would recommend? Maybe a bit philosophical. Um, I mean, there's some really good books for cultivate. If you're into like foraging, so if it depends what area of mycology you're into. If you're into cultivation, then like the psilocybin Bible is a really really solid. It really covers loads of the low tech methods that are applicable to all areas of mycology. There's also obviously the old Stamets books like what's it called? It's called I don't know. It's gone. Growing gourmet mushrooms or something. But it's one of the Paul Stamets books. It's a bigger one. That's got like all these like old school bits in them as well. Um, maybe a bit philosophical. Could mycelium communicate with others? Like there's an argument that that mycelium is communicating with us, or in some respect, by getting us to talk on its behalf. Like I mean, I'm like I said, I'm here because of a series of life-changing psychedelic experiences. So I guess there's an argument there that mycelium is communicated to me and then now continues to communicate through me. And that, yeah, maybe that's a bit woo, but I like that idea. Why do you pick the mushrooms before the veil breaks? It's a purely aesthetic thing. So there's no issues. So spores generally will not germinate on the surface of like mycelium that's already colonized. So it's more the fact that it just looks ugly. So As the veils break, they deposit spores onto the mushrooms below, and it can create like a really mucky looking end product when you're looking at the final mushrooms. So yeah, normally it's good to just kind of get things right just at the right point. My rice is not breaking and shaking in my jar. So you can sterilize, get like a metal. Someone earlier was saying about using a metal. You can buy metal chopsticks and you can sterilize those or put them in like boiling water, and then you can use that to break everything apart in the jar, and that should get it out. What you don't want to do is like whack the glass jar against things because yeah, it's a recipe for like ending up in A&E. Mate, I've got to say that you oh, thank you for T McDonald said, mate. We got ADHD that was executed perfectly. Thank you. I'm just in a hyper. So I've had like some medication today for my ADHD as well. So and a couple of drinks as well. So it helps. What about cordyceps? Right. So cordyceps is like so to give you an idea of like cultivating of cordyceps, it's there are people that specialize in just doing cordyceps. There is so much to it, and the cultures are so the selection for good cultures of cordyceps is so such a like a fine art, and requires so much discovery in like a breeding sense that you do end up like with people who will just specialize in that. So cordyceps earlier on I was talking about culture senescing, where the genetics just like immediately tire out through just the creation of new cells, and they like human beings I guess they just get old and tired. Cordyceps does this very quickly, so you cannot so like it can't be stored well. It doesn't store well. It doesn't reproduce well. But I think that the actual findings now is that it's not so much to do with senescence, of like this tiring of the genetics. It's more to do with there being a more dominant part of the organism that tends to wipe out the opposing mating pair. And I'm not 100% on that, but that, from what I can tell, is where the science is moving now. Is that basically like there's some part of the organism that basically kills off the other part of the mating pair, or maybe it's more dominant. And as that gets transferred, that dominant side tends to take over, and it becomes just a weaker genetic organism. Um, you need to research that yourself though. But yeah, cordyceps is like if you want to dedicate your life to something mad, like there's a couple of guys in the UK that are doing cordyceps. Lawrence from Kaizen Cordyceps, who like is just dedicated to just doing cordyceps breeding. And so he gets people all over the country to send him cordyceps that they found in the field. Um, Steve asked, what products and MycoPunks can be used for microremediation and how? You can use, so you could basically create grain spawn and add that to, so there's a company called Fleet Field Lab. I don't know if they're a company or like an organization. Fleet Field, they create grain spawn of like of sporeless oysters. They transfer that to these big sacks of pasteurized straw and sawdust, and then they use the mycelium in those bags to remediate or clean up like contaminated soil. So oyster mushrooms are really cool in the fact that they break down carbon chains. So you can give them quite complex like contamination. You could give them like a cigarette butt, something that's made out of like all sorts of like different polymers and plastics, and it will break down that stuff and till there's just nothing left. And the same with crude oil. You can feed it petrol. All these just really horrible contaminants that we have in our environment. Oyster mushrooms will break that down, and the mushrooms that are produced from this remediation are even edible, which is crazy, and they don't have any of the original contamination in them. But yeah, you could buy an oyster culture and some grain spawn, and then just like literally just get sacks like Hessian sacks full of substrate, allow it to colonize, and then you can use that to remediate soil. And there's obviously a bit more to that, but yeah, if you follow Fleet Field Lab, they're called, and it's spelt like this. I think it's Fleet. Oh no, it's Fleet like that. Field Lab. Um, all right, so to use a knife or just twist the fruiting bodies off when harvesting. I tend to like just lever them back and forth. So like, I like to just because I think when you cut them, you end up with like you just end up with loads of little stumps of like gooey mushrooms at the end. So I think leaving them off and taking a little bit of the substrate with it is fine. And then I tend to just clean the substrate off after. But yeah, that's generally the better yeah, the cleaner way of doing things. Um, no worries. And yeah, Steve, the remediation stuff is really interesting. Again, like this is another area of mycology that like people in the UK are now starting to, to move into. And I love that. I think, people are starting to find their niches, find where they feel comfortable within the wider mycological picture. And that's kind of where like, where MycoPunks is in this community is like I want to support all those people who are finding their niches. And that's where that's where I see MycoPunks being is like we want to just support people finding you know, I want everyone to just quit their jobs and just and work with mushrooms, and I want to support that in any way I can. Um, so yeah, yeah. Um, anyone got any more questions? I don't know how many people are even left. I missed the beginning of the broadcast. Yeah, it is going to go on YouTube. Yeah, so it is being recorded. Um, so yeah, it'll all be on here, and also there were some technical issues as well that I hope will be solved by the end recording. So fingers crossed. But yeah, um, Steve, the golf course farm. Yeah, yeah, exactly. So this can like farmland, anywhere where there's heavy machinery being used. The old coal spill sites. There's all sorts of sites that could do with remediation. Most of the UK is arable land, so the farmland that just needs the soil fixing. There's a place for mycelium in that picture to take us back to a better better habitat. Um, Small said thanks. Thank you very much. For anyone who's like filtering out, by the way, thank you so much for joining. Like it's been really cool. It's nice. Um, it's nice to chat to everyone, even though it's not like a very two-way with just me chatting at you. Does a flow hood filter require ISO sprayed on it prior to use? So no, and actually like it's something you really want to avoid. Um, so the way these filters work is they're like a very delicate paper filter that has like a that just has like a specific pore size to it. So it's like imagine if you were trying to blow air through a piece of tissue paper. Um, it's very delicate. So when we're working in front of a flow unit, we try and avoid spraying anything onto it because any liquid, even isopropyl, has like a wicking mechanism where as it dries, it draws things back into the filter. So what you can end up with is like the liquids will effectively dry after you turn off your flow unit. It will draw contamination onto the paper. So yeah, the paper is like clean. What we normally say is like when you turn these on, you just run it for like 5 minutes before you use it, and that will launch out any surface contamination. Or I've had like you end up with spiders living in there sometimes, which is obviously when they're not on, it's a good little place for a tiny spider to go. So. But yeah, they don't need to be sprayed with anything, and you definitely shouldn't. So, when's the next 4 for 3? So we're trying to like taper back a bit on doing deals that aren't related um to like loyalty. We're trying to like I want to take a like step back from just doing plain discounted stuff and start offering some things just for like people who are part of our Discord community and like I want to do like secret products that like no one else has access to that might combine like a bunch of different things that might be good for a weekend project. So, the idea I've got at the moment is like doing like a like an agar pack, a bunch of like little things for you to get stuck into some new project with clearly like defined instructions to it. And then that'll be like a box that is cheaper than it would be if you bought all the things separately. But yeah, I think the problem is, as a business, like you can end up like with people waiting around for the discounts. So you struggle with cash flow throughout the month. So it's like, yeah, it's an annoying business thing, but yeah, and people come to just expect everything to be discounted. So I want to kind of move really to like rewarding people who are like, who like are part of our community. So. Um, Joey, thanks so much. Really useful. Enjoyed this a lot. Me too, mate. It was really cool. Yeah, thanks so much for coming. Any other questions? Any new products in the pipeline? Uh, yes. I'm working on we're working on a way of converting our top down our flow units to a top down flow unit. So basically, like a stand that will let you just work underneath like a top down flow unit in like a cabinet with with perspex sides. So it will allow people who've bought like the small flow units and stuff to have like a much bigger area to work under that's much more useful. So yeah, it basically lets you turn like the mini flow unit, which is the one I've got in front of me here, into like something that's really, really useful. Not that it isn't really useful now, but it opens up the possibilities a bit more of what it can be useful. And we're also just in the final stages of getting press like the equivalent of a Presto in. And we've been working with a Dutch company that has a C marked Presto, like version of a Presto, which is slightly bigger. Um, it's got all the safety certification, and it's just really good. Like, and they've considered mushroom growers in their development process. So there's like the stuff within it is all like designed for people like us. So that's another product that I'm hoping to kind of get on sale soon, which will hopefully just go up for pre-orders and then we'll have it in like as a general. So yeah, like a pressure cooker. So the pressure cooker that we're looking at, you could, for instance, so a pressure cooker is like for anyone who isn't familiar, we talk about all the sterilization of different things here. Um, you can make your own grains, you can make your own grain spawn, make your own agar. You can do all these things using a pressure cooker. It opens up a world of possibilities for you after a still air box. A pressure cooker really is the next most important thing that you could do to become more self-sufficient with your growing. It means you don't have to buy, grain off us. And I really want to encourage people to like, while like, we're running a business, I kind of want to wean you off some of our like more beginner products and like, get you making stuff yourself and actually getting you really, more involved in the process. What's the staff Christmas MycoPunks staff Christmas party gonna look like? Do I need some lasers? Um, we are going to a place called Beefy Boys, which is good for all the people who eat beef in our organization, which is about half of them. They do some good vegan stuff as well, yeah, but yeah, we're going to a place called Beefy Boys, and then we're going to go out and just drink a lot, I think. Um, but yeah, no, I think there won't be any need for lasers in a restaurant, but thank you. No, I'm saying Instapot. Yeah, so it is like an Instapot, but it's much bigger and it's and also the cool thing about this is it works on induction, so you can basically like set it and forget it once it comes up to temperature. You can like put a timer on and leave it for 2 hours and come back and just have grain spawn ready, which is very cool. Um, Will my grain spawn jars dry out with the humidity at 60%? The warmer my pantry, the drier they seem to get. So, so once are you talking about when they're fully colonized or like I mean the humidity is generally quite low anyway in like in warmer environments, but um yeah, I mean unless you're seeing like an issue with it, I wouldn't worry. It's Yeah, I mean 60% is not too bad. Yeah, it's not too bad. But yeah, if you see an issue with it, then you could always just put a couple of open jars of water in with stuff, and that'll just bring the humidity up slightly. Like the ambient humidity, I mean. Susanna, it's an awesome talk. I believe I completely messed up my current project, but we live and learn. Like look, if you're having problems with any like niche problems, anyone who's not on our Discord community, you should absolutely join it. I'm going to put it in chat now just so you've got it. It is literally just myco.chat. So that's our Discord community. There's over 5,000 growers in there, of all different levels. Our team are active in there. We've got like moderators in there as well who are like high level, growers who help people out in detail with any problems that they're having. And like, yeah, I'm in there, the team are in there. This is a really good way of getting some hands-on help. I think like in the first year of when we started, like I tried to help everyone in a really like individualized way, and it's kind of what MycoPunks was built on is like mad energy I had in the first year and giving everyone like individualized help. Now when you've got 25,000 customers, it's not something that there's anything other than a recipe for burnout. So I try and like push people towards like helping each other out rather than me just jumping on a call with everyone. But I wish I could help you though on and on everything. Um, yeah. Has anyone else got any questions? I'm around, I'm just around as long as you want. So, God, that's so good. Right. I'll give you that. Oh, thank you so much. Listen to the whole night and not get, but oh, that's good. That's really reassuring. Look, it's been a really nice. Oh shit, there's actually a Q and A bit here that I completely missed. That's funny. So we were just obviously doing everything in chat. I'm gonna answer some of these questions that were in the questions and answers as well. Right, Joey, I've grain spawn, but it has brown stuff at the bottom. So that could be either bacterial. So if it looks sludgy and like milky, then that could be bacterial. If it looks like like a lighter color, like almost like a lighter brown crust, or like a powderiness that's over your grains, that could be like a yeast thing. If you want to send me a photo on support, I can literally give you even if it's not one of our products, I can give you my feedback and let you know. Um, so you also asked, can you clone two traits from different mushroom bodies on one agar, for instance, one bit of white mushroom and a bit of spotty mushroom at the same species? No, so I don't think cloning works in that way. I said earlier that like cloning is uh, so cloning you're basically like I th I don't know. I think I'm going to end up like trying to shoehorn an answer into this. I don't believe so because I think that like the mycelium that you get from cloning is not the same as like what you would get at the early stages of germination. There's a lot more going on there. But you could try it, like, I mean, you could definitely try it. Once you know where, I mean, I guess I don't see why it wouldn't. Maybe I don't know. I'm willing to say I don't know. So yeah, answer that one. Done. Um, See, we talked about white rice as grain spawn earlier. Like you can do that, and you should definitely give it a try. Have a look at Uncle Ben's tech. Um, I used it to put blue oyster gnocchi on for new. So is the kit for gourmet mushrooms or is it not recommended? Noam said so. So our like our beginner grow kit, it's a good way of you trying out the process of growing gourmet mushrooms, but it's not an ideal substrate. If you have a look at, we have a substrate called Rise Above, which is like a wood-based substrate. If you're getting serious about like growing gourmets, then I would look more towards the wood-based substrates than coir. But coir is a lot more forgiving when it comes to contamination. Um, right, one more question in here from T McDonald. Something I could hardly find any info on was using UV boxes for sanitizing. The nubby little travel bottle sterilizer we're knocking about. Would that kill spores and mycelium inside a syringe? So yeah, so UV will damage like any organism basically. Like it if you left spores under UV, then they're likely to get damaged. Um, I think from what I can tell, like dark spores especially, if I remember rightly, dark spores especially are like more damaged by UV, whereas clear spores, maybe I may have this the wrong way around. Clear spores are like they were. No, actually, I have got that the wrong way around. So dark spores are more suited to being in UV. That's where you see them more in mushrooms that are in fields. Tend to be a lot more dark spores, and the whiter and clearer spore stuff you find in like shaded areas. So the color of the spore has a lot to do with how much built-in UV protection it has. But yeah, I would not be using UV sterilization for anything in cultivation really. It's like we have like a UV light that's built into one of our overhead flow units, not the ones that we make. But like, and it has you can turn it on before you leave the lab, and it will kill bacteria. But like if you're doing stuff at home, it's just you'd be more better off just using normal clean technique like isopropyl spray uh, and just like wiping surfaces down with bleach and stuff like that. Um, but yeah, I wouldn't bother trying to use any UV stuff. There's a really horrible story that happened a couple of years back of someone working in front of a flow hood with a UV sanitizer in it, and they basically like just burned their eyes out like really badly, damaged their vision just from working in front of UV without realizing it was on. Um, to create liquid culture from spores, do I follow the same steps that you did with the LC to agar? Yeah, so you can either have a look at. I'm going to just quickly get the instructions from. Let me get the instructions for the agar pots. Um, so there's both a video and like a if you go to the easiest fucking agar pots, there you go. So this actually covers the liquid culture stuff as well. If you're someone who prefers watching videos, then there is a video on our YouTube channel as well, which is uh, mycopunks.com. No, youtube.com/mycopunks. So there's videos on there on how to use the agar pots as well. If and some people like reading like step-by-step guides, and some people just like watching a video. Um, cool. Anyone else got any more questions or no, thank you, Simon. Thanks for coming. Really appreciate it. And yeah, I'm really glad, I kind of like I didn't know that's good to hear, Steve. Like I didn't, know how it would go, whether it would be technically too difficult or but I really like the idea of just being able to just guide people through using some of our products and just some of these techniques. And it'd be really good, if you guys end up trying stuff, I'm really interested to see your results. So we're on Instagram as well and stuff like that. So just tag us in a post or send me the photos. You can send them on support. Suzanne, lovely to meet you. Thank you so much, and yeah, let me know how you get on with your agar because I'd be interested to see. Thanks, no. Thank you, Ellie. Cheers, Lee. Right, appreciate it. Cool. If there's no more questions, I'm going to give like another 30 seconds. I think everyone's filtering out now. What PS5 game are you going to hit? Battlefield 6. If we're on Battlefield 6 now. It's it's become like it's the one thing I do to blow off steam in the evenings. Like normally I'll come back, I'll have a bit of food, do a bit of work, and then I'll play PS5 for a bit with people. Cheers, Steve. Thanks for coming along as well. I appreciate it. Um, right, the number's dwindling down now. We're at 15 people. What are you waiting around for? Cheers for the Mushroom Community Hub. Thank you, Morad. Yeah, and thanks for coming along. Appreciate it. Now it's gonna be like one of those no you hang up type moments because it's the whole 15 people. I'm just gonna carry on drinking. Thank you, Joey. Thanks so much, Carly. Really appreciate it. In fact, I think you might get weirdly, you guys might get an invite to review the actual Mycology Club product that like I guess technically you bought by coming here. So yeah, leave me a review if you want. That'd be cool. And yeah, I'm almost certainly going to do that. Well, I'm definitely going to do this again. Well, I think it works really well. It's a cool format. But it would be good to do like some sort of something that's a bit more interactive. Right, we're on 13 people. I'll make myself another drink. Any other questions? 12 people. I feel like I got to be the last one here as it's my thing. 10 people. Just leave the computer on. That's funny. Right. I'm just going to be the one to cut the thing off, and feel really bad now. If someone's writing like a really long question, and then I disconnect. Here we go. That's funny. All right. It's basically a lunchbox with UV lights built in. You zip it up, then turn it on with a power bank. I thought it could be good for sterilizing equipment in a home environment. So if you're talking about like scalpels and things like that, that you're trying to sterilize, you can actually just get like a little pot of isopropyl, like a little just dish of it, and just keep it, sat in that isopropyl for a while. That's a good way of just sanitizing stuff that's generally enough, enough for most stuff. Um, there's also flame sterilization. I think all of those things are going to be are going to be better and less risky than, that's all right, less risky than doing like a UV bath because also obviously UV is only going to get one side of things, so you're going to need to open it up, turn it all around, which is going to be a nightmare. There is also the fact that you can just boil stuff. You could if you've got metalware that you want to use over and over again, or syringes, the needles, you can just boil them in a pot and then take them out in a clean environment. So. Oh, we're down to six people. Right, off you fuck. Come on. Right, I'm gonna just leave now. Um, I need to have something to eat. Nice to see you. Thank you all so much for joining me. See you later, buddy. Bye. All right, take care. See you later. Bye.